So Melody, what are the top three challenges when developing a cell and gene therapy?
Well, thanks for asking the question, Alex. I think the first one for me would be the manufacturing of these cell and gene cell therapies. And that is mostly because they're such individualized treatments nowadays, makes it very difficult to have them in mass production. And one example would be CAR T cells. And as of now, we have to actually at the bedside of the patient, take the cells out of the patient, get it to the lab, get the CAR T cell in there and then transfuse back into the patient. So that makes the mass production very, it doesn't work at the moment. And the off-the-shelf version of these CAR T cells will be a huge step forward. But I think the second challenge, I would definitely say cost of goods. And that is mainly due to the fact that most cost of goods models that are applied to gene and cell-based therapies are based on conventional biologics. The difference is that, for example, gene therapies, they are aimed at fixing a genetic disease, a genetic defect, and potentially being a one-time treatment. And then, of course, having a biologic that is administered often and in some cases even chronic. These are models that are not applicable for the cost of goods of these cell and gene therapies. So we need to develop new models in order to make them fit for purpose. And with the limited data that is there at the moment, because there are a limited number of patients that have received these novel therapeutics, or therapies, it is going to be a challenge in the future to have enough data to actually build such a model. And then the third of these are the assays around how do I make sure that my product is pure, how that my product is safe, that my product is good to be administered into the patient. And for example, the CAR T cell, how do I make sure that I can administer it back after I have changed the T cell into the CAR T cell? Well, the thing is, with potency assays, that is one of the assays used to identify strength and stability of biologics. You do apply that to gene and cell therapy as well, but because it is such time sensitive, but also unique material, it's very difficult to find good reference material. And I think clinical data that we get from the patient over the years will feed back into what is an adequate in vitro assay, but we are not there yet. So it will take some time, but that is one of the other challenges for the development of cell and gene-based therapies.